Just go ahead and pick up where we left off on Friday. Lesson that we had kind of discussed were the structures that make up the cytoskeleton. And that brings us to the point now where we can start to talk about other stuff with the cell. We'll get to talk today later about the plasma membrane, which forms the outer boundary of the cell. Okay. The cytoskeleton in some cells will push out on that plasma membrane to form these projections that provide special features and functions to certain cells. Now, last time we talked, we learned about the cell size, and we got to see perfect cube-shaped cells, okay, where all the plasma membranes were nice and flat for the 6 sites. Not all cells are going to be like that. Some cells will have what we call microvilli, found in epithelial cells that line our intestines and other areas where we absorb. Absorption is where stuff is brought into the cell. Okay. We can see here is a cell with several microvilli. This singular form of microvilli is villus. These are these little finger-like extensions, and what they do is increase surface area. Surface area, in this case, just basically means the amount of membrane. Okay. They can do so up to 20-fold. They're formed from bundles of microfilaments. What they enable a cell to do is have more space and more places for stuff to come in, which is what absorption is, or exit, which is what secretion is. And the second type of extensions are called cilia, little hair-like extensions, 7 to 20 or 10 microns in length, composed of microtubules. They move in a rhythmic, coordinated manner to push stuff along the surface of the cell. It's important to understand that in our body, we have them in places like our respiratory tract, where we need to move something along from cell to cell, basically. Like in our respiratory tract where we push contaminated mucus, like mucus that's collected dust that we've breathed in, push it upward to either be spit out, coughed out, hacked up, or down into our digestive tract to be digested, okay, in our stomach. And we also have them like in female fallopian tubes that push the egg along. They don't move the entire cell, they just move something along the surface of the cell. Okay. Sperm, however, move the entire cell, like in or like flagella in sperm, which are kind of like a whip-like tail that help a sperm swim and implant or fertilize, a female egg. So those are 3 extensions of the plasma membrane. Now, associated with some of those are our first organelle, what we call centrosomes, which are organelles located near the plasma membrane or near the nucleus, made up of some kind of specialized stuff. I'm not too concerned with that. I do want you to know their functions. To anchor and assemble microtubules, which if you look at them, they actually are formed from microtubules. They help move chromosomes during cell division, and they aid in the movement of flagella and cilia, which we just talked about. Okay, so that's really kind of all I want you to know about these guys, those 3 little functions. Now, if you take an anatomy, or you're currently taking anatomy, which some of you are, this is a nice review, okay, because we've talked about it before. So I'm going to be able to kind of buzz through a lot of this stuff. If I get going too fast, though, don't hesitate to holler at me. Our next organelle is the same name as something that we've seen before. Back when we talked about atoms, we learned that the nucleus of an atom is where you find protons and neutrons. And, well, in the nucleus of cells, we find DNA, which we've talked about before. DNA being a nucleic acid, and is our heritable stuff that we get from mom and dad. For that reason, we describe the nucleus as the cell's information center. Okay, and nucleus is a lot like the plasma membrane, which we'll talk about later today, and that it has a double membrane that surrounds it, which is called the nuclear envelope. That helps to contain the DNA. Now, in any non-dividing state, cells are going to have their DNA kind of in an unwound state. It's still going to be in a double helix, but it's not going to be tightly coiled up into chromosomes. Instead, we describe it as chromatin. You can see it here on the next page, which is this kind of grainy mass of DNA, but you also find RNA and proteins. When cells prepare to divide, DNA will wind itself up around these proteins called histones, coil up, and eventually organize into chromosomes. Why? So that they can duplicate their chromosomes. And so when the cell divides, each cell, because you go from 1 to 2, will have a total of 46 chromosomes. 23 that came from mom, 23 that came from dad. And that is what our life starts out with when we have the formation of zygote occurring. 46 chromosomes that have come, half from mom, half from dad, eventually dividing, all of which later on in life will contain that same number. Now that does not include our sex cells that we produce in adulthood, which in men are sperm produce 23, females you're going to produce 23 in your eggs. That way if you reproduce, you're going to produce a zygote with your partner containing 46 chromosomes. Overall, the function of the nucleus is to control all cell activities because of the instructions contained in DNA. Remember, last week we talked about the various organic molecules. We learned that if something is happening in the body, who can we assume is doing it? What type of organic compound? Was it carbohydrates? What? Proteins. Okay. Now, we just said the nucleus contains DNA. Remember the other day I wrote up this DNA makes RNA, which makes proteins. This is why the nucleus is the control center, because it contains the DNA that ultimately ends up with the making of the proteins. The making of RNA actually also takes place within the nucleus. We're going to see that is going to take place in the middle of the nucleus in what's called the nucleolus. Okay, and then the other function of the nucleus is just storage of genetic material like I was just talking about. The nucleolus is kind of like a secondary organelle located inside of the nucleus. It's non-membranous, so there's not a membrane surrounding it. I would highlight this sentence here, or just really this part right here, which is its function. Synthesizes RNA from a DNA template. Okay. That RNA then combines with proteins to form the subunits that will eventually form these things called ribosomes that we'll see on the next page. One of the most distinctive features of the nuclear envelope, that surrounding membrane, is that it's double layered, and that sprinkled in throughout are these nuclear pores, which is how RNA can escape the nucleus to then be used to make proteins with the ribosomes that surround the nuclear envelope. So you can actually see ribosomes. These are little kind of grayish dots on the outside of the nuclear envelope. We find a lot more ribosomes on the RAR. We'll talk more about those here on the next page. And it's pretty cool how we form these nuclear pores. It's all about proteins and fusing together of the nuclear envelope. I'm not really going to get into it, but it's really important because it allows only certain things in and out of the nucleus. And it prevents stuff that shouldn't get into the nucleus from getting in, and only allows RNA and other couple of things in or out. So it's very selective in what can pass through. The next organelles are called ribosomes, which you can think of as protein factories of the cell. We find them kind of all over the place. We just saw some on the outside of the nuclear envelope. Primarily, we find them attached to what we call the rough endoplasmic reticulum, which was actually pictured here on the previous page. And what ribosomes are very small collections of more than 50 proteins and a type of RNA called ribosomal RNA or rRNA that when you combine its large and small subunits completes the functional ribosome, which is not super important. Okay. We also find ribosomes just kind of floating around the cytoplasm. We call those free ribosomes. We find them inside of our mitochondria. We call those mitochondrial ribosomes. We'll see that here in a couple of slides. But the primary place we find them is attached to our next organelle, which is called the endoplasmic reticulum, which we saw a couple of slides ago. It's kind of a stack of membranes that extend throughout the plasma, or cytoplasm, excuse me. Those membranes are tubular, and inside we have a lumen, which is where freshly made proteins and fats will accumulate, or calcium will be stored, because sometimes we find that some endoplasmic reticulum stores calcium. And the ER is continuous with the outer membrane of the nuclear envelope, therefore the space between the two nuclear membranes is the same as the lumen of the ER. There are two different forms of ER, smooth and rough, which are named because of their appearance. Rough looks rough, smooth looks smooth. You can see that really nicely here on the next page where we're showing off both rough and smooth. Okay. RER, as it's sometimes abbreviated, most of the time you'll see me abbreviate it that way, is rough in appearance because of those many ribosomes attached to it. Now, since RER has ribosomes, and ribosomes are the protein factories of the cell, overall the function of the RER is to synthesize proteins. What happens is pretty cool. The proteins will accumulate inside of the membranes in the lumen, and then, as you can kind of see here, the membranes will kind of pinch off and form these little bubbles, which are going to transport those proteins from the RER to the next organelle that we'll see, called the Golgi. And these little bubbles are called transport vesicles, kind of like little vehicles. Okay. Now, since the SER does not have any ribosomes, the SER has no role in protein synthesis. It does, however, make fats, things like fatty acids, steroid hormones, cholesterol. Okay. Those are all made here. SER is also responsible for inactivating, detoxifying certain chemicals. Harmful things like drugs and alcohol, pesticides, carcinogens. You guys know what carcinogens are? Just any chemical that can cause cancer. And asbestos, you hear about it. And your liver cells are responsible for performing these functions. Okay, so like habitual drug users or alcoholics, if you look at their cells in their liver, going to have a whole bunch of SER because they have to be always working. Okay. SER is also involved in carb metabolism, calcium storage. We'll talk a lot about its role in calcium storage when we look at muscles. Okay, we actually changed the name of the SER when we talk about muscles. It's now known as sarcoplasmic reticulum. And it releases calcium. Without calcium, our muscles won't contract. We'll get to see that in about a month. So we'll look at that function of the SER later. Before we move to our next organelle, I want to talk about gene expression just briefly, which I've already written up on the board with our steps from DNA to RNA to proteins. This is an easy way to describe gene expression. Although cells synthesize many chemicals to maintain homeostasis, much of our cellular machinery is devoted to synthesizing proteins in large numbers, which we know proteins can be thought of as responsible for most everything in our body, and therefore they determine physical and chemical characteristics of cells, therefore us as the organism. It begins with transcription, which is where we take DNA and make RNA. Transcription. So we convert from a DNA template to an RNA. Translation then is where we go from RNA to proteins. RNA attaches to a ribosome. Information contained in RNA is translated into a sequence of amino acids forming a protein. Remember how we talked about amino acids kind of like letters of the English alphabet? How if you arrange them in specific ways, just like letters in a word, you can get all sorts of different words, which all have different meanings. Hey, I like that last sentence there because it kind of helps tie that in. Conversion from the nucleic acid language to the protein language is another way to describe translation. Transcription takes place, like we said, inside the nucleus, thanks to the nucleolus, which is not shown in this image for whatever reason. But we go from that double-stranded DNA to RNA. RNA that then leaves the nucleus through those nuclear pores and combines with ribosomes, is read and made into a protein through translation. If you take biology, you're going to learn about these in much, much greater detail. Okay. All of those processes, both of those processes, are very intricately controlled. Okay. Here, we're just really explaining what they are. Okay. Now, those freshly made proteins, or fats, if we're talking about the SER, are going to then be carried to the Golgi. The Golgi is our next organelle, sometimes known as Golgi apparatus or body. I just call it Golgi because that's an easy way to remember it and describe it. Also looks like a flattened stack of pancakes with membranes and lumens. Okay. These guys alter those proteins and fats that were made by the ER. Here on the next page, we can show you what I'm talking about. I like to think of the Golgi kind of like FedEx or UPS, which receives packages when you drop them off, modifies those packages to get them ready to be shipped off, and then ships them off and delivers them. So if you think of yourself as delivering a package, you drive to the UPS place to drop it off. Okay, that's like a protein getting carried from the RAR to the what we call entry phase or cis face of the Golgi. This is the face that always is pointing towards the RAR or SER. Okay, if this was SER instead of a protein, it would be a fat. Okay. So you drop it off and then your job's done. Okay, but then they alter the package, probably slap a couple stickers on it, maybe put a barcode or two on it, scan them, whatever. That's what the medial Golgi does. Okay. There are such things as glycolipids and glycoproteins. We're actually going to talk about those here in a little bit. Be like a sugar attached to a fat or a protein. Glyco refers to sugar or carb. So it is basically a carb protein or a carb fat. Glycoprotein, glycolipid. Okay. Or we can, like I said the other day, with proteins have to have a specific shape. Okay. A lot of times the altering of the shape can take place within the medial. So that's where things are processed, put together into their kind of final product. Okay. The shipment of the protein, glycoprotein, fat, glycolipid, takes place by way of the exit phase or transphase, which when you're thinking about a package getting delivered, how's it getting carried? By way of a vehicle which we call transport vesicles, just like the transport vesicles like you drive in the vehicle or driving your package there. Okay. Now, let's think. We said the other day, like hormones are produced, hormones which could be a fat, which could be a protein. Hormones are released into the bloodstream. How do they get in the bloodstream? Well, we show that right here, exocytosis. We'll talk about that process next time. That's basically where that vesicle fuses with the membrane and spits out the substance inside. What if it's a phospholipid? We said phospholipids are the main components of the plasma membrane, right? Well, it might fuse and become part of the plasma membrane. Instead of phospholipid here, they're calling a protein. That's merging with the plasma membrane. We'll talk about membranes that are part of the plasma membrane here in a minute. Sometimes we form transport vesicles that don't leave the cell, but end up staying inside to become a functional organelle, like lysosomes. So overall, the key function of the Golgi is to package different molecules together, can be used elsewhere in the cellular body. Well, let's talk about those lysosomes that I just mentioned. These are kind of like the garbage disposal cleanup crews of a cell. They're only about 0.5 to 1 micron in diameter, and they're formed when that singular little bubble breaks off of the Golgi. So it's a single membrane, and inside of it are as many as 60 digestive and hydrolytic enzymes. Basically, it's these really strong chemicals that can break stuff down. Hydrolyze, remember, means to break down. They can hydrolyze or break down proteins, nucleic acids, complex carbohydrates, and fats. They can digest, dispose worn-out organelles, like mitochondria, for example, wear out over time. That process is called autophagy. Auto means self, phage means to eat, so it's like self-eating. It's pretty cool how it works. The organelle basically gets surrounded by a membrane formed by the ER, and then that bubble combines with the bubble of a lysosome, forming what's called before that it forms an autophagosome, and then fuses with the lysosome, and then the lysosome digests it. So that's kind of cool. Foreign material can be degraded, brought into the cell, destroys the entire cell in some cases. Autolysis is what that's called. And then one of the coolest ones is what your sperm do, guys, if you want to make a baby. They release their hydrolytic enzymes out of the cell altogether, so they perform extracellular digestion, and that's how a sperm can penetrate a female egg, what's called an oocyte. So that's kind of cool. We'll actually talk about that process way later in the semester. That's actually the last lecture, and towards the very end of that lecture at that. So we'll talk about that in May. Long time from now. Organelles that look like lysosomes but differ in function and are a little bit smaller are peroxisomes. Inside of them are several oxidases, which are enzymes that can oxidize. That's the name, what's oxidases? Oxidize means to remove hydrogen atoms from, and so these guys remove hydrogen atoms from organic substances. We find a large number of these in our liver. Another name for peroxisomes are microbodies. What they do is degrade hydrogen peroxide, which is a potentially toxic compound. Neutralize free radicals. We talked about free radicals. Remember I compared those to like Tasmanian devil. Those are those molecules or atoms with an unpaired electron. Just really reactive. Cause cancer. So we want peroxisomes to neutralize free radicals. Detoxify drugs and alcohol. So just like SER, they do that. And then they can also decompose fatty acids. Proteasomes, there's really not that much discussion on those. In fact, when I was in college, you guys' age, I don't ever remember learning about these guys, so I don't know if they've been just recently discovered or just kind of ignored. But these guys kind of destroy unneeded, damaged, or faulty proteins by cutting them into smaller peptides, which, remember, peptide is just a smaller chain of proteins, or amino acids, excuse me. Our last organelles are mitochondria. Probably my favorite. We know them a lot of times as the powerhouse of the cell. which if I were to ask you guys, maybe you do or don't know what that means. You will here in just 1 second. We find them in all eukaryotic cells. We are eukaryotic organisms. It means that, well, I'm not even going to get into it, but that includes us and other animals and plants and fungi. What they perform is aerobic respiration, which we learned what the word aerobic meant the other day, or what it means. What does aerobic mean? With oxygen, and we learned respiration refers to energy production. So this is where that oxygen that we breathe in goes to produce our ATP. For that reason, we call them the powerhouses of the cell. The more active the cell is, like muscle cells, the more mitochondria you're probably going to have, because they require more energy. Structurally, as you can see here on our next page, mitochondria have a double membrane, just like we saw with the nucleus, and just like we'll see with the plasma membrane. The outer mitochondrial membrane is the least important of the two. The inner is important because if you look at it when we see it here from the inside, you'll see that it folds in on itself. That inner folding of the inner mitochondrial membrane increases its surface area, which, there's that phrase again, means that we can have a whole lot of enzymes. A whole bunch of enzymes. These enzymes are important in that production of ATP. If you've ever taken biology and learned about the electron transport chain, if you haven't learned about that, you will later in the semester. That's what these enzymes are involved in, which is where most of the ATP production takes place. This inner folding of that membrane is called the mitochondrial cristae. Now inside of that is a fluid called the mitochondrial matrix, which is where the Krebs cycle or citric acid cycle takes place. Which if you never learned about that, we will talk about that towards the end of the semester. So we'll talk a lot about mitochondria later in the semester. We'll talk about it throughout the semester at various points as well. Also within the mitochondria, you'll find its own DNA, which we inherit from our mom, which is pretty cool because we inherit mitochondria from mom. From dad, we really just get their nucleus because sperm are basically just a nucleus inside the head and then the tail. Okay. So mitochondrial DNA does code for different things, just like the DNA that we have in our nucleus. So that's kind of cool. There's also ribosomes. That's what these little blue dots are. So that's kind of cool. We talked about those earlier today. But yeah. All right. What we're going to do now is transition to the outside of the cell and talk about the plasma membrane. That outer boundary, which we learned about the other day because we talked about the primary structures that form it. What we describe as those phospholipids, which were unique fats because they were amphipathic. Amphipathic, remember, means that they have polar and nonpolar regions, which in our drawing here of a simple phospholipid, is it the head or the tails that are polar? The heads. Now, since they are polar, they do something different than most water or most fats do with water. They are attracted to it. What was the word for that? Do you guys remember? Hydrophilic. The tails are just like most other fats in that they are nonpolar and therefore are water-fearing or hydrophobic. We talked the other day that when you put phospholipids together in watery solutions, they're going to form a double layer, which we describe as the lipid bilayer. Now, if you look at the image at the bottom, you find that there aren't just those phospholipids, which are the kind of turquoise heads, and then the kind of the gray tails pointing inward. We also find proteins, like we see here with a channel protein that's labeled. We also find glycoproteins, which are proteins with a carbohydrate. We find things like cholesterol, which is a form of fat. We even find glycolipids, which are lipids attached to fats. Primarily, our cell membranes are made up of phospholipids. But the carbs and proteins provide for other functions, and this ratio between these components is dependent on the function of the cell. Let's discuss those functions and structures of the plasma membrane. The overall model to describe the structure and arrangement of lipids and proteins in the plasma membrane is described with what we call the fluid mosaic model. It's important to understand that the plasma membrane is not like the walls of this room made up of these cylinders, these big blocks that are bonded together and are very rigid. Okay. The proteins and fats that make up the plasma membrane are nice and dynamic, and that they can kind of float around and move around. And it's not structured and rigid, but it is put together and does help define the outer boundary of the cell. It's just a lot less rigid than like the walls of this room. You can actually tag by highlighting, kind of an easy way to describe it, various proteins and lipids. And at one point, you might see a lipid or a protein on one side of the cell, and then later come back and it will be floated off to the other side. It just shows that they can kind of move around a little bit. Let's first talk about the three different types of lipids. We already discussed one, those phospholipids, which are the primary structures, making up 75% of the lipids in the plasma membrane. You've got 5% that are glycolipids, which we saw a picture of one of those on the previous page. Glycolipids, remember, are carbs attached to a lipid. Okay, glyco for carb or sugar, lipid for fat. 5% of the lipids here in the plasma membrane are this type. We only find them in the extracellular space, so they only point outwards. They help to have cells adhere to one another, so stick together. May mediate cell-to-cell recognition and communication, and provide for cellular growth and development. That leaves us with 20% of the fats, which are cholesterol molecules. Talked about cholesterol the other day as a steroid. These provide strength to the plasma membrane, but also decrease its flexibility. So that is kind of a drawback to them. We can kind of see them sprinkled in within those tails. They're kind of these yellow streaks sprinkled in with the tails of the phospholipids. Proteins of the plasma membrane are classified as either integral or peripheral, which describes their location within the plasma membrane. Integral extend into or through the bilayer. Most of them are described as transmembrane. So a lot of times you'll see integral and transmembrane kind of used interchangeably. Transmembrane means that they span through both layers. And so they poke into the cell, into the side of the cell, and poke out into the extracellular fluid. Many of these are actually glycoproteins. Not all, but many of them. Which means that they are a carb attached to a protein, just like we saw with glycolipids. Together, the carbs from both glycoproteins and glycolipids creates this sugary coat we call the glycocalyx. From one cell to another, this pattern of carbohydrates will vary, which actually allows cells to recognize one another, okay, which is important because cells have to communicate through a variety of means. Okay, we'll talk about that a lot this semester. This helps cells adhere to one another, protects them from being digested by enzymes in the extracellular fluid. The other type of proteins are called peripheral proteins. We either find them pointing inward or outward, but they don't extend through it. Okay. Here on the next page, we can talk about them in a little bit of greater detail, and so we'll do so right now. Some integral proteins form what we call ion channels, which are kind of like the door, really more so the door frame that we pass through to get into this room. Okay. Because the phospholipids have those tails that are nonpolar, not anything can just pass directly into or out of the plasma membrane. Just like we can't just pass directly through this wall like a ghost could. To get in and out of this room, we have to go through the doorway. Okay. As we come in and out of this room, that door, and really the door frame or doorway, does not change its shape. Okay. That's different than how the next guys work. Carriers or transporters physically change their shape, allowing substances to come in or out. Okay. Glucose comes into your cells this way. Okay. Ions, more than typically, will use a channel. Okay. And we have specific channels for specific ions. Meaning like sodium only passes through sodium channels, potassium only passes through potassium channels. They're selective in what they carry. Okay. Channels also can sometimes have a door on them. We call those gated, just like this door that's needing to be open to allow us to come in and out. Okay. We'll talk a lot about these this semester. And we're going to talk a lot about movement across the membrane here today and next time. The third type there are receptors. Integral proteins serve as cellular recognition sites. Each type recognizes binds a specific type of molecule known as a ligand. A lot of times ligands are hormones or neurotransmitters. Okay, great example of this is on our thyroid gland. There are receptors for a hormone that's literally called thyroid stimulating hormone. Okay, thyroid stimulating hormone might reach the entire body, but will only bind to these receptors for thyroid stimulating hormone. Okay. And then the thyroid will be stimulated. So, very important. Allow cells to target one another. Enzymes, we talked a lot about those the other day. Integral or peripheral proteins catalyzing, which means, once again, speeding up chemical reactions at the inside or outside of the cell, or plasma membrane. Linkers, which can be either integral, which are the yellows, or the peripherals, which are the purples. Anchor proteins of plasma membrane and neighboring cells to one another, work with protein filaments inside, outside of the cell. And then membrane glycoproteins, glycolipids often serve as cell identity markers, kind of acting like a flag on top of a castle, allowing cells to recognize one another. These are important, like when we talk about white blood cells, how they can recognize if something's foreign or not. Unfortunately, sometimes white blood cells will attack our cells, and they can cause different diseases. We call those autoimmune diseases. If you've ever heard of like multiple sclerosis or type 1 diabetes, rheumatoid arthritis, we'll talk about how the immune system's kind of failing us in that situation. So. Now, I've already mentioned some of these various functions of the plasma membrane. You can see there's a figure there showing off them as well. Just like the walls of this room, the plasma membrane helps define the boundary of cells and organelles. It defines the room or space within it, like the walls of this room defining us in here versus out there next door. Plasma membrane also helps to organize the internal organelles. Just talked about how the plasma membrane has these various proteins that act as receptors, so serving as sites for receiving signals. Signals that could be like a chemical, like a hormone, could be an electrical signal, could be even a pressure signal. Okay. Chemical signals could be like the things that you taste. There are cells with specific receptors in our taste buds. Taste buds are found in your tongue. Okay. When we taste stuff, there are chemicals in our food that stimulate those receptors and then send the signal in, and our brain processes that, and we decide, hey, that tastes good, or it tastes bad, or it tastes salty versus sugary, or meaty instead of bitter. We'll talk about taste and other senses like that later in the semester. Pressure can be as simple as like pushing in on your skin. And there are pressure-sensitive receptors associated with neurons in your skin that allow us to, feel touch. Okay. And then receptor sites, or those proteins, like I said, to help us stick cells to one another. And you can kind of see over there on the right. What we'll talk about the most, though, with regard to the plasma membrane, is its function to move stuff in and out. Possessing transport proteins, facilitating, regulating movement of substances into and out of the cell. This is important, as it states, transport of materials across the plasma membrane is essential to the life of the cell. Certain substances must move into the cell to support metabolic reactions, while other substances produced by the cell must be moved out. The last slide that we'll probably get to today is this that just begins us into what we'll talk a lot about on Wednesday, which is cellular transport. These terms might be brand new to you. If they're not, great. If they are, that's okay. Term permeable and impermeable are opposite one another. Permeable means that a structure will permit the passage of substances through it. Impermeable means that it won't. So the word permeability refers to whether or not something passes through. And so we describe the plasma membrane as having selective permeability. What do I mean when I say that? Only certain things can pass through it. Think of the plasma membrane kind of like a dish strainer, okay, where if you're making like macaroni and cheese, you dump the water and the macaroni and cheese into the colander or strainer. Water passes through, but not the noodles. Okay. The lipid bilayer is highly permeable, meaning that it can easily pass through to nonpolar, uncharged molecules. Things like O2 and CO2 can go directly through with a big old arrow to illustrate it. Okay, it'd be like if we could pass directly through this wall. Doesn't need anything because they can go directly through. It's unaided. Okay. Steroids also are highly permeable, or the membrane is highly permeable with steroids. Remember what steroids are? Fats, right? Fats can generally pass right through other fats. And remember what makes up the primary structures of the plasma membrane? Phospholipids, which are fats. Lipid bilayer is impermeable, won't pass through. So ions, sodium, potassium chloride, get rejected. Other things that are large and uncharged, like glucose, how do they get in and out? That's where those channels and carriers, like the door that allows us in and out of this room. Lipid bilayer is slightly, there's a smaller arrow here, slightly permeable to small uncharged polar molecules like water and urea. For the most part, though, we can basically assume, because of those tails that don't like water, water is kind of more like these two guys, charged and large things, so they have to use proteins. And we'll talk about that in greater detail on Wednesday. As we pass into cellular transport, we'll pick up with this slide where we talk about passive and active on Wednesday. You guys all have a fantastic rest of your day.